Hello everybody, welcome back to the next IB Biology G4 Lab video. In this video, we'll be covering the topic of microscopy, including subtopics such as types of microscopes and their components, microscope calculations, and the different types of techniques you can use on microscopes in order to pass your IB Biology HL exam. First of all, let's discuss the different types of microscopes and the components within a microscope. Now, there are two different types of microscopes that exist. First of all, there's the light microscope. You've probably seen them in your previous science classes, but this is probably what you envision as a usual microscope. Then there's the electron microscope, which is a lot more complex and basically a lot more detailed and more expensive. Now, covering the light microscope first, one feature that they have is they can see color, which you've probably tried out in a previous science lab. Not only that, they're able to see movement and live cells under their lenses. And they're not too much of a high cost compared to the electron microscope, and they're able to see up to around 1500 times magnification. In terms of resolution, it's about 0.25 to 0.3 nanometers, and we'll discuss what those words mean later. Now, magnification is one word you do need to know pretty early on, and it's how much bigger a projection looks compared to the actual subject size. So for example, you can see in this picture here we have a smaller A, but through magnification we're able to see the A much more clearly and much bigger. A formula you need to know along with the magnification is the total magnification formula, which is the ocular times objective. Now the ocular is one component of the microscope which is near the eyepiece, and the objective lenses are the one you switch around for higher magnification. Resolution is the smallest measurable interval from a projection, and it's the quality of how visible a projection is associated with. So for example, we have a high resolution cube on the left, while lower resolution, more blurrier cube is on the right here. And the last term we'll cover here for now is field of view. It's the total diameter of the projection. And in this image I have made here, it's supposed to mimic what's under the microscope and what you usually see through the ocular. So for example, this total field of view could be around 1 centimeter if you set it to be that. Now let's cover the electron microscope. Now the features of the electron microscope is that it cannot see color, it cannot see movement and live cells, and they have very high costs, all of which are disadvantages. However, the advantage of electron microscopes come with the fact that it has around 300,000 times magnification and around 0.001 nanometers of resolution. Now, an important note when using microscopes, especially a light microscope, is that the projection will be flipped and upside down from what it is viewing. So for example, this letter E here will look like that under a light microscope. Now let's go into some microscope techniques. Now microscope techniques are probably what most of the microscope questions on the IB exam will look like, and there might be a few calculation questions on it as well, but let's cover the microscope techniques first. Now for the IB exam, there are three key techniques that you want to know: the freeze fracture, the immunofluorescence, and the cryogenic electron microscopy technique. Let's cover immunofluorescence first. Now this is the technique and process done for light microscopes that involve binding chemicals with color that can be seen as bright images. This technique allows for moving parts and proteins of cells to be tracked and viewed easily because, of course, if they're brighter and they can be seen easier, it's easier to track them and look through the microscope. Now let's get into cryogenic electron microscopy. Now this is a process done on electron microscopes that involve freezing the cells and hitting them with a bunch of electron beams. Now this technique allows for the internal structures of materials and whatever's under the microscope to be examined with much more depth than, on a standard just non-technique electron microscope. And then finally we have freeze fracture. Now this is a process done on electron microscopes as well that involve freezing the cells and fracturing them along weak points, as the name would suggest, obviously. And this technique allows for internal structures of whatever is under the microscope to be able to be examined, like different membrane structures. Now let's start off with some microscope calculations. This is most likely going to be on the IB exam in one way or another, and will definitely be on your unit tests if you are being taught this unit at school. Let's start off with some FOV calculations. Let's say we're given that the magnification here is 40 times and that each interval here is 1 millimeter. Here, because we can clearly see 3 marked intervals here of that 1 millimeter, we are definitely sure that there are 3 millimeters here. But because there's a little bit of space in the left and the right, we can put a guess in for the next unit down, which would be the point or the 10th place of the millimeter. And because it's a little bit of a millimeter and not too much, I put down the guess that it would be around 0.2 millimeters. So in total, I guess that the FOV is 3.2 millimeters, which through the IB examiners, they'll probably say is correct. Let's take this one step further. Let's say we up the magnification from 40 times to 100 times. We will try to find the FOV of the magnification, or we'll try to find the FOV of this projection at magnification of 100 times now. Now, one formula you'll definitely need to know is the FOV times magnification equals FOV times magnification. Now let's input the values into this equation. Now we know that the original magnification was around 40 times and that the FOV was around 3.2 millimeters. So we can input that into the left side of the equation to make 3.2 times 40, and then set the other side to FOV times 100 since we're trying to solve for 100 times magnification. Taking this step further, it will be 128 equals FOV times 100, and we could divide the 100 from one side to the other, and then we get that the FOV at 100 times magnification is around 1.28 millimeters. Okay, let's do another problem with a different calculation, which is the size calculations under the microscope. Now let's say we're given that the FOV here is 1 millimeter. And for these types of problems where you try to calculate the size of individual subjects under the microscope, you're going to need to do a bit of an assumption. So here I said around 2.7 cells fit within the FOV here. I mean, there's some 3s somewhere, there's 2 of them somewhere, but I think around 2.7 cells within the FOV is a pretty fair assumption here. Now the calculation we'll want to do is FOV divided by the number of cells that fit, which should be around the estimate size of each cell. So here, since we were given that 1 millimeter was the FOV, and there are 2.7 cells within the FOV, we can conclude that each estimated cell size is around 0.37037, which is around 0.4 millimeters. Now we'll probably be getting into the most important formula of them all, which is images and magnifications and actual size. Now, I like to call them size calculations, and this is an image version. And let's just say we are given that the magnification is 200 times and that this image is 3.7 millimeters. Now the important formula you need to know is image equals magnification times actual size, and this is interchangeable in a way which would be like A equals I over M and such, but this should usually be given in your formula sheet, but you should just memorize this formula because it is probably going to be the problem you get on the exam. Let's go back to our problem here. We can identify our variables here, which the image is 3.7 millimeters and that the magnification is 200 times. Since the unknown variable here is actual size, we'll be trying to find the actual size. So let's put our formula down first, I equals MA, and then we can derive that actual size is image divided by magnification. Let's input our values here. The image is 3.7 millimeters and the magnification is 200. And then if you calculate that, we can find that the actual size is 0.0185 millimeters. And then for sig fig's sake, it is 0.02 millimeters. Now let's try a problem on your own. Let's say that the image is 50 millimeters, and the unknown variable is the magnification, and the actual size is 0.9 millimeters. I'll give you a second to work on it. Okay, the actual answer you should have gotten here is 60 times. The magnification should be a 60 times variable or somewhere around that, but you should be getting 60 times. Now this is the reminder I'd like to give you. These are the two biggest formulas that I'd like you to have in mind. Image equals magnification times actual size, and FOV times magnification equals FOV times magnification. If you need any more help on any of these questions, or if you're confused on how I got 60 times for the last answer, please leave a comment down below. In our next topic, we will most likely be discussing cladistics, so please stay around. Thank you for watching.